Hello everyone, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we're going to be talking about evolution and the theory behind it. Thanks for being here. And so before we get too far into this, there are basically two types of evolution. There's microevolution and macro. Micro means small, macro means large. Microevolution is the type that is observable, and you can see these small scale changes happening over a shorter period of time. An example, just like we're seeing here, let's say you've got a population of mice and they're different colors, and there's a predator in the environment. And that predator is going to be able to find mice of one color a little bit easier than another. And you can see over time, the color of the mice shift. And so what's happening there is an example of microevolution. This is the vast majority of examples that you're going to see in textbooks and people talk about when it comes to evolution. These small scale changes that can happen over a shorter period of time that generally are contained within a species. Now, macroevolution is the type that we typically think about when we're using that word. Evolution means change over time, and macro is large, and so these would be large-scale changes over longer periods of time. It's assumed that microevolutionary changes will accumulate over long stretches of time, hundreds of thousands, millions of years, and lead to all the diversity that you see today in life, from molecules to man, evolution, and everything in between. Now, Charles Darwin was the person who is widely credited with coming up with this theory of evolution in the 1800s, and he didn't do it on his own. But one of the things that he did is he said that there is this thing called natural selection, and that is when you have a population of diverse organisms, some of those organisms are going to survive at a higher rate than others. And so maybe they'll be able to exploit food sources or avoid predators a little bit more efficiently, like we just talked about in the previous example. Well, those offspring, the ones that are best suited for an environment, are going to leave behind more offspring. And in his day, he had artificial selection, which was human beings that would select for certain traits, and then those organisms would leave behind more offspring. But that was an artificial selection because human beings were dictating which organisms survived and reproduced and which ones didn't. But in natural selection, it's natural forces. There could be predators, there could be disease, there could be environmental changes. It's important to note that in natural selection, it cannot select for the perfect organism. There are limitations to what natural selection can do. It can only act on existing populations, on existing genetic pools, and natural selection is such that what's optimal at one time may not be optimal at another time. So this is a nice example of natural selection. In the upper left-hand corner, you've got a population of bacteria. Some bacteria are resistant, some are susceptible to an antibiotic. When you add the antibiotic into the environment, the resistant cells are going to be the ones that survive, and pretty soon you're left with a population of just drug-resistant microbes. It's one of the ways that antibiotic resistance occurs. Now, it's important to note here that when you have these changes in a population and when you have these genetic mutations, it's always a compromise. And so usually the drug-resistant microbes are not going to survive in an environment as easily as the microbes that are susceptible when the antibiotic is removed. And so it's important to understand that these mutations that happen are always compromises at best, and usually they're going to be detrimental to the microbe or to the organism that experiences a mutation. In other words, it's going to be bad for the organism when they experience a genetic mutation. So Charles Darwin did not come up with this idea on his own. In fact, the idea had been out there 100, maybe 200 years, if not longer, before he ever popularized it. He just came up with a framework by which it could potentially work. But he was influenced by other scientists around his time. One of them was Lamarck. And what Lamarck thought was kind of interesting, and that is he assumed that as organisms exploit different sources, it's going to change them physically. And those physical characteristics that they change over time, or that they develop over time in the course of their life, will be passed down to offspring. So here's a really good example. So Lamarck thought that as organisms grow, as they get faster over the course of their life, as they get stronger, in this case, as a giraffe reaches for higher and higher food sources, well, those physical characteristics, as they develop, may be passed down from one generation to the next. So another really good example is, let's say you happen to be going to the gym a lot, or you are a sprinter, or you are a bodybuilder, or a weightlifter. As you gain those physical characteristics, those would eventually be passed down to the offspring. And that turned out not to be true for the most part. except for one important thing. Recently, scientists have discovered this concept. I say recently, but certainly within the last 20 or 25 years or so, we've discovered that there's this thing called epigenetics. And in a previous video, we learned all about genetics and how traits are passed down from one generation to the next. But here, what happened was we've got this situation where in epigenetics, what's happening is quite interesting. The genes are actually changing their structure, not the sequence information, but their structure inside a nucleus. And those structural changes can be passed down from one generation to the next. They can be accumulated over the course of a lifetime, and they can be due to certain stressors, and it can impact the structure of genes, which then impacts how the genes are functioning. But what's more interesting is that those types of structural changes can be reversed from future generations, even though they're passed down. Alfred Russel Wallace was another person who influenced Darwin, but in a different sort of way. He actually came up with the theory of evolution through natural selection around the same time, and he just said, hey, I'm going to publish all of my thoughts and findings. And Darwin was like, whoa, time out. You know what? I've been working on this now for decades. I just haven't gotten around to publishing yet. And back in the 1800s, I think science was a little bit more civilized, and Alfred Russel Wallace allowed Darwin to publish his thoughts and his theories before Alfred Russel Wallace did. And so Darwin is rightly credited with the theories before Wallace. So Darwin was influenced by Lamarck and the fact that there was change over time. He was also influenced by Malthus. And what Malthus said is he wrote a very important book in the 1800s and said, hey, you know what? A couple things are going to happen over time. One thing is that populations are going to continue to increase. And they're going to increase to a point where they're going to populate an area more than the food supply can handle. And at that point, there's going to be a struggle for existence. And only the best organisms suited for that environment, sound familiar, are going to be the ones that survive. In other words, the organisms that are able to acquire food in space are going to be the organisms that survive. And the ones that are weaker, well, they're not going to be able to eat food, have a supply of food, they're not going to have a territory, and they are going to die out. And so those ideas heavily influenced Darwin and his theory of natural selection. Another person who influenced Darwin was Charles Lyell. He was a geologist, and what he was doing is he was studying geological processes and how long they take. And one of the things that he studied was sedimentation rates, the amount of time it takes for sediment to build up in layers. And when he started to calculate this out based on the rates that he was seeing, he was one of the first people to popularize the idea that the Earth was maybe older than a lot of scientists thought. And at the time, the Earth was assumed to be around 6,000 years old. He said, wait a minute, these sedimentation rates are taking a lot longer. Based on the number of layers I see, the Earth could be tens of thousands of years old. Now, of course, he made some assumptions here. And the assumption is that the sedimentation rates were the same throughout history as what he was witnessing. And that's not something he could possibly know. But most scientists just assumed, okay, whatever the rate is of sedimentation or processes that we see today, we're going to assume that they've always been the same in the past. But again, that is an important assumption that really can't be scientifically proven. Now, based on that, when scientists started to look at fossil records, what they found was that organisms, generally speaking, that were simpler were deeper in the fossil record, deeper in the sediment, and organisms, as they became more complex, were closer to the surface. But there are a lot of important examples where that doesn't always play out. But when it comes to the fossil record, what we should be seeing is, if evolution is true and there's change over time, simple things towards the bottom. As you get closer to the surface, more recently, you should see more complex organisms. So during the 1800s especially, there was a lot of debate because there were two competing theories. One was catastrophism, and catastrophism is basically what we see today is the result of many major catastrophes over time. Things like floods, volcanic activity, asteroid impacts. These things would have a very massive impact on ecosystems worldwide. But once Lyell came along with Darwin and all the scientists that were in his group, they believed in what is known as uniformitarianism, especially Charles Lyell, where he said, you know what? As time passes, you're going to get a fairly consistent rate of sedimentation. And later on, that was applied to radiometric dating, something that we're going to talk about before. So catastrophism, you had these major global events that impacted how we view our ecosystems. Uniformitarianism, It was a slow, gradual change over time. Now, as I've already alluded to, there was a problem with the fossil record. And when Charles Darwin was coming up with his theory of evolution, the fossil record was far from well studied. They really didn't know the parts and pieces of a cell. They didn't know about what made up a genome or genetics or anything like that. But one of the things that he said was, hey, listen, if my theory is true, then what we should see over time is transition. We should see transition from simple organisms to more complex, and we should see transitionary fossils that tie everything together. We fast forward about 100 years, we get in the 1960s, 1970s, and the fossil record was lacking a whole lot of evidence for that. And so scientists were trying to figure out, why is that? Stephen Jay Gould and Niles Eldredge, back around in the 1970s or so, they came up with this theory of what is known as punctuated equilibrium. And what they said was, when you look at geological time, what happens is you've got a whole lot of boredom in between vast periods of time. That was the equilibrium part. And so you have things that basically go on unchanged, because they notice that most of the fossils that you see appear in the fossil record fully formed, and they're in the fossil record for a period of time. And if they leave and go extinct, they basically look exactly the same as when they first appeared. And so what they said, to try to explain things like the Cambrian explosion, where all of a sudden you've got a whole lot of nothing and boom, pretty much every single phylum that has ever been known to exist just magically appears in a very, very short geological time period. So what Stephen Jay Gould and Niles Eldredge said through punctuated equilibrium is, you've got these long periods of boredom followed by these very short periods of dramatic change, and that could actually be due to catastrophes. And so in some ways they kind of circled back to and started looking more at the catastrophic events that could lead to dramatic changes, like the Cambrian explosion, where you see a lot of organisms buried in the fossil record all at one time. Now, their ideas, of course, open up a whole lot of other potential issues and questions, a little bit too much for us to get through this introductory video. So one of the ways that scientists try to date things around them is by using radiometric dating, or isotopes. And these are elements that tend to break down into other elements over time. And some of these break down at different rates. And so you're going to see things like uranium up here at the top that have a half-life, the amount of time it takes something to lose half of its original concentration. You can see here we've got billions and millions of years. And carbon-14 is super popular. We talked about that in a previous video. Carbon-14 is an isotope that breaks down, but it takes it roughly 5,700 years for half of its original concentration to go away. But you can see that there are other elements that break down much faster. And so you've got fluorine, breaks down in about 110 minutes. And so that's its half-life. And so the amount of time, once again, it takes for half the original concentration to basically be converted into something else. And so here's a really good example of how to think about this when it comes to half-life. I'm going to use some arbitrary numbers here, but let's say you have element X, and it's an isotope, and it's going to break down, but it takes 200 years for it to break down into half of its original concentration. So 200 years is its half-life. And so we're starting with 100% at time 0. 200 years later, you'd have half of the original, or 50%. You'd go another 200 years, and you'd have 25% of the original, or a quarter. You go another 200 years, you have 1/8 or 12.5%. And every time you cut that number in half, it's a 200-year difference. And so if you had something that was started out with, assuming it started out at 100%, and it went down to 12.5%, you would know, based on this example, hey, you know what? The thing that we're looking at is 600 years old. Now, just like with anything, when we're looking at uniformitarianism, there are some important assumptions you have to make that really can't be tested that easily. And when it comes to radiometric dating, there are 3 basic assumptions. Assumption number 1 is that this half-life of 200 years is consistent. In other words, there's no opportunity for the half-life to speed up or slow down, or anything that has happened environmentally or ecologically that would cause that half-life to change. Because if it does, that would throw off our assumptions and our numbers. And so you think about it like an hourglass. If you had sand at the top of an hourglass and it was falling to the bottom, we can't have a situation where the hole that allows the sand to fall gets wider or more narrow over time based on environmental conditions. The second thing that we have to assume is that nothing has been added or taken away from the system. In other words, our hourglass is sealed. We can't have sand being added to the top. We can't have it being removed from the bottom, because if that happens, then the whole time situation really doesn't work. And so when it comes to radiometric dating, we have to know that our parent element was in fact 100% at time zero, and our daughter element is building based only on the half-life and the conversion from parent to daughter. And finally, we also have to assume that our conditions are known at the start of the time. So in other words, we have to know exactly how much sand was in the top of that hourglass. And of course, that is an assumption. All three of these things are assumptions and things that we cannot possibly test, because we're dealing, at least in terms with longer periods of time, there's not one single person that can test that over time. Now, we can get that information from the radiometric isotopes that decay at a much more rapid rate, but for the ones that decay at a longer rate, these assumptions just have to be made when using this method for dating things. All right, so the next thing we have to make sure we identify, and that is the concept of a species. And a species is a group of organisms, when found in nature, will produce fertile offspring. Now that gets a little bit tricky, because sometimes you have species that will be not necessarily found together in nature, but when they do meet, they create something called a hybrid. And that's a mix of the two species. And so you have different relationships between speciation. The first thing that can happen is something called reinforcement. You have a group of species, and as they separate, they become more and more unlike one another and less likely to produce hybrids, and over time they become two completely separate species. But you can have something called stabilization, where you have a group of organisms that do begin to separate into two different species, but still have a fairly vibrant hybrid zone, even though you have speciation occurring. And then the last thing is something called fusion, where you have a group of organisms that begin to diverge from one another, begin to speciate, but then something environmentally pushes them right back together into a single species and a single population once again. Now, it's really important to note there's a confusing concept here, and one is gene flow. You want gene flow between the members, the individuals of a population, because that's going to maximize genetic diversity. That's a good thing. However, for speciation, you actually need gene flow to stop, and you need reproductive isolation. In other words, you need two groups from the same species to separate so that they can eventually, over time, and if the environments are different enough, become two separate species. So while gene flow is good for the health and genetic diversity of a population, you need to stop, you need reproductive isolation to happen in order for speciation to occur. And speciation can happen in a couple of different ways. I'm using African cichlids here. These are fish from the Great Lakes of Africa. There's lots of different species. There's some hybrids. And when it comes to species of African cichlids, the males tend to be really, really colorful, and the females are usually like a brown or a tannish color. And it's the males that have to show off and do the dances and show off their fins and show off their colors to entice the females to mate. Now, there's two different types of speciation. One is allopatric, and that means the two species are separated in space. They don't generally meet up, and therefore they're not going to create any fertile offspring. The second one, really important for African cichlids, is sympatric speciation. In this case, well, the fish are all intermingling. They're all mixed, kind of like what you're seeing in these fish tanks, but they generally ignore one another and don't create hybrids because the males are so different that the females really don't pay attention to the males that look different from the males that they're normally attracted to for mating. Now, keep in mind there's a couple things that can speed up the process of speciation, and that is, there's two concepts. One is the bottleneck effect, and I have this picture here. Imagine that you have some type of a catastrophe. You had a big population of organisms with a lot of genetic diversity, and then something happens, and we dramatically reduce the number of individuals within that population. Well, there's a very high likelihood that the genetic diversity of that population, because there's fewer organisms, are going to be less genetically diverse than the original population. And that, in turn, can lead to a group of organisms that starts to take on physical characteristics that were not necessarily representative of the larger population as a whole before the catastrophe happened. So that's the bottleneck effect. And there's another one that's very similar. Now here, this is called the founder effect. Imagine that you have a land mass and you've got a big diversity of ants. Well, a small group of ants wind up on a log, and that log floats across a big body of water to an island where those ants don't exist. Now, maybe in the original population, you have millions and tens of millions of these ants with an amazing amount of genetic diversity, but on this log, turns out there were only 5 or 6. But they survived, they made it across, they had their voyage, they inhabit this new area. Most likely, the new group of ants in the new area is going to have a genetic diversity that is very different from the original population. And over time, especially if that environment differs, the new environment differs from the original environment, you could have a distinct genetic diverse population that could become a new species. Another thing to consider is that natural selection is not stationary. I mentioned this already. What is advantageous in one environment for one population, as the environment changes, can become, well, less advantageous. And so sometimes you'll see species that change their microevolutionary characteristics through natural selection over time. And so there's 3 ways that that can happen. One is stabilization, and that is the environment surrounding that species is basically making sure that we have less diversity in terms of physical characteristics. So I'll give you a really good example. Let's just say we've got mice. We have black mice, brown mice, and white mice. And we have a situation where we have a stabilizing effect where it's just the brown mice tend to survive, those ones right in the middle, they tend to survive greater than the black mice and the white mice do. So that would be stabilizing. But we could also have a situation where we have disruptive selection where there's something that is consuming, maybe a predator is consuming the brown mice. Maybe there is some genetic situation there where the brown mice are more susceptible to a parasite, and the black mice and the white mice are surviving, but not the brown mice in the middle. And so that would be disruptive. And then there's something called directional speciation or stabilization, and that would be, in our example, we have the black mice and the brown mice and the white mice. Maybe one of the extremes is being selected over the others. And so maybe there's more snowfall, and the white mice are blending a little bit better, and the black mice and the brown mice, they're getting picked off by predators a little bit more efficiently. And so now we're starting to see that shift towards the white mice. Now, it's important to understand there are 2 main types of barriers when it comes to whether or not speciation is going to happen. Prezygotic and postzygotic. Before, zygote means the formation of a fertilized egg. Prezygotic means that sometimes you can have organisms that are found in nature together. However, they're not going to be able to produce offspring. And so when it comes to prezygotic barriers, there's a number of them. One of them might be habitat. So you're seeing here we've got a snake, and this snake is spending most of its time in the water. There may be lots of snakes on land all around this particular snake, but they're never really going to come into contact because even though they're in the same general area, they inhabit different habitats. This snake would prefer to be in water. The other snakes would prefer to be on land. They're really not going to meet up, and therefore we're not going to see a whole lot of mating between this snake and one that would be on land. You could also have something called temporal isolation. Again, this is prezygotic. You're never going to have fertilization because maybe what we have here is we have animals that are going to be found in the same habitat, but they're active at different times, or maybe even different times of the year. And so you've got nocturnal animals, you've got animals that are awake during the day. And if they were potentially around and active at the same time of day or at the same time of year, they might be able to form fertile offspring, but because they're active at different times, they don't. Again, that's a prezygotic barrier because they're active at different times. Another potential prezygotic barrier is behavioral. And so in nature, it's kind of interesting because a lot of times we're dealing with fish or birds, it's the males that have a lot of color. They're constantly showing off and they're trying to attract females who tend to be not as colorful. Well, in behavioral isolation, sometimes the female doesn't like the color of the male, and so especially we talked about this with cichlids, you have females that are usually going to be like a brown or a tan color, and the males have all the color. When they're trying to show off, if they're the wrong color, the females will not mate. When it comes to birds, sometimes they have to do a dance, they have to show off their feathers like the peacocks do here, and if the females don't like that dance, they don't like the colors that the male is showing off, there's not going to be any mating there. So that's an example of behavioral isolation. You also have something called mechanical isolation, and that is the parts just don't fit together, male or female. An example I like to use is, imagine here, that is a female toy Chihuahua, and you've got a male Mastiff. That's not going to work. And so the parts have to fit together, the reproductive parts have to fit together, and if they don't, there's not going to be any opportunity for new offspring. And same thing with gametes. You have to make sure that when two organisms, when they're mating, or at least nature figures this out, if the gametes don't work together, in other words, maybe there's a different chromosome number between the gametes of two different organisms, there's not going to be an opportunity there for fertilization. All right, well, what about post-zygotic barriers? Let's say we have two animals, they meet in nature, they reproduce, and they actually produce offspring. Well, in this case, there are some examples of post-zygotic barriers. One would be you produce hybrids like this liger here that really is infertile. It will not reproduce after its own kind. And so you've got these post-zygotic barriers where you have infertile offspring, hybrids. Some hybrids are going to have reduced viability, and that means as time goes on, as more hybrids are produced, they just kind of break down over time. And then there are other types of hybrids where they're just going to have reduced viability, and that means they don't make it to reproductive age. So the last concept I want to talk about here is once we've got all of these organisms, human beings are trying to categorize them, classify them, organize them in a way that makes sense for us. And that is the job of taxonomists and phylogenists, where they make these phylogenetic trees. And basically what they're trying to do is group organisms based on, it used to be physical characteristics, but more and more now we're looking at the genetic makeup of organisms, which often leads to physical characteristics. And so genetically, we try to determine which organisms are most like one another, which organisms are most dissimilar, and then we try to group them in basically a family tree. And we've talked about this in a previous video, but this is basically the classification system. It's missing one really important one, and that is domain towards the very, very bottom. There are domain, which there are 3 of them. And then, as you can see here, we've got these categories. And as we go from bottom to top, we get more and more specific. We start narrowing down until we get to a species, which is that individual organism I talked about earlier in the video. And so we do have ways, we do have these classification systems, but know that it's very, very difficult for us humans to deal with all the diversity that we see in life, to classify them. It's a big job, and it's not necessarily easy to do. All right, everyone, so that was a brief overview on how things change over time and speciation and what leads to it. As always, thank you so much for being here. Have a great week, everyone.